This is a nice way to start out talking about kidney function. What we see here are a couple of different examples of kidney stones. The one on the left is considerably smaller. The one on the right obviously is larger, but they also have a little bit different structure to them. The one on the left is going to be a little bit more lumpy. The one on the right is going to be a little bit more filled with sharp serrated edges and projections on it. But either way, both of these are relatively large stones in the grand scheme of kidney function. Really what happens with kidney stones is that perhaps the kidneys aren't flushing enough fluid. And if they don't have enough fluid flushed through, then buildup of waste products can occur. So one problem might be chronic underhydration. There might be some other things that would be associated with kidney disease. That means the kidneys aren't very good at flushing things on through the system. But either way, if this occurs, it can seriously block up the system. And if it lodges somewhere, obviously cause a backup of urine, an increase in pressure in the kidneys. Obviously can be very painful as it's passing through, and especially if it becomes lodged. And if it's really big, there are a number of things that can be done about that. But I'll leave that to your reading assignment materials. But this is just an example of what happens when the kidneys don't work like they're supposed to. Here's the overall system, and it starts with the kidneys. So the kidneys produce urine. The ureters take the urine from the kidneys to the bladder. The bladder holds the urine, and then the urine passes through the urethra to the outside of the body. And that's the 4 big steps in the process. But by far the most complicated is the functionality of the kidneys. So let's spend our time looking at kidney function. Here's a slice through some kidneys, and what we see here is that we have a renal cortex out here around the edge, outer edge or outer surface of the kidney. Then we have the medulla, or more the middle part of the kidneys here, and both of those are going to be involved in the process. And we'll get into the idea of nephrons here in a little bit, but over here are some nephrons, and here's the outer cortex. So the nephrons and a fair bit of the actual filtering structures of the kidneys are found in the renal cortex. And then the nephron loops and most of the collecting ducts are found in the medulla. And all those things will make sense here in a little bit. So actual filtering of urine occurs out in the cortex, and then, or maybe we should say formation of urine, and then the concentration of urine happens in the medulla. And that'll, again, make sense here in a little bit. So what's going to be filtered out? What is filterable from the blood? And obviously water is filterable, as are nitrogen-based wastes, nutrients, and salts. And some of those things we wanted to take out, and some of them we didn't. So we'll find out here in a little bit that the kidneys are actually too good at doing what they do, and they have to undo themselves to a great degree. Some things that are not filterable that are in the blood are the formed elements. So that's going to be the red blood cells, white blood cells, and platelets, and some of the plasma proteins. And really what determines what's filterable and what's not at this point is dependent on size. So the formed elements and some of those plasma proteins are simply too big to fit through the filter. And we'll take a look at that in a minute. I mentioned that the kidneys do too much. They're too good. So here's what happens. If we look at water, and we can see the amount filtered per day, around 180 liters. That's a lot. You don't have that much in your body in the first place. So what that means is that most of that water has to actually be reabsorbed. So the kidneys take it out, but then the kidneys have to put it back in before it becomes urine that's excreted. So of those 180 liters that are filtered, less than 2 of those actually become urine typically, with a reabsorption rate of 99%. Sodium has a fair bit filtered out, only a little bit of it actually excreted. It's actually reabsorbing 99.5%. Glucose is filtered, but it should not be excreted, and if it is excreted, that's typically diagnosed as diabetes. So you get 100% reabsorption there. But urea, that's the waste product, actually has about a 66% excretion rate. So not even all of the waste product that's produced is actually excreted, and part of the reason for that is it helps to regulate water and salt balance in the system. But again, that's of these four considered here at least is the one that is excreted the most. So let's look at this process. And we're going to start the process with the glomerulus. So here we find the glomerulus, this little capsule structure. And over here in this image, each of these would be a glomerulus over here. And so there are loads and loads of these in the kidneys. And this is where urine formation starts. So the blood comes in, runs through this pressure, and this is basically a pressurized filter, runs through that filter, and if the pressure is high enough, it forces stuff out into the glomerular capsule, into the space around the edges here. And then that blood that is now cleaner goes out of the glomerulus and back into circulation again. From the glomerulus, it's going to go into a series of tubes. The first one here is the proximal convoluted tubule, and that name sounds a little bit convoluted too, but it's simply saying closer tube that is twisted and convoluted. Proximal means closer to, and convoluted means twisted all about. So it's a tube that is closer to, in this case, the point of origin of the glomerulus and is twisted all about. Then it goes from there to the nephron loop. You might occasionally see this listed as the loop of Henle. It's the same thing, and the nephron loop here. And then towards the end we have the distal convoluted tubule, and distal simply means farther away from. And then the distal convoluted tubule will end in the collecting duct. So we got a couple of different points here at which the urine is being processed. And of that filtrate, and the filtrate is the stuff that's filtered out by the glomerulus. So anything that is removed from the blood by the glomerulus is a part of filtrate. And so filtrate consists of water, nutrients, and salts, and obviously waste products as well. And of those, most of the water is reabsorbed, most of the nutrients are reabsorbed, hopefully all of them if we can, and most of the salts are absorbed. And what's not absorbed, in other words, what actually becomes final product urine, is some of the water, hopefully most of the waste, and any salt that is extra. So let's look at that process. And I already said it's a pressure process. So blood comes in here, goes through these tubes, basically these capillaries, and these capillaries are covered in filtering tissue, which allows some things to go through and retains the non-filtered stuff. The red blood cells, the white blood cells, platelets, and those proteins we said were too big, they don't get pushed through there, then that blood exits. So that's the filtrate, everything that squeezed through. The filtrate then goes to the proximal convoluted tubule. And remember, we've lost water, we lost salt, we lost nutrients, we lost waste molecules from the blood into the glomerular capsule there. And then so all that stuff goes here into the proximal convoluted tubule. And in the proximal convoluted tubule, we're going to start reabsorbing those things. Remember, most things got reabsorbed to some degree. So we're going to reabsorb salt, water, and nutrients. That's going to be the three main things that get reabsorbed by the proximal convoluted tubule. And this is the only place where nutrients are reabsorbed to a great degree. And so that means that the sugars, the fats, the proteins, that sort of thing, need to be reabsorbed here. And if the proximal convoluted tubule is not able to adequately remove the sugar, that's where you actually get glucose in the urine, is because there was just so much of it in the blood that the proximal convoluted tubule couldn't take it all out. Not necessarily that the proximal tubule wasn't doing its job, but there was just so much that it could only do so much because it only had so much opportunity. It's a relatively short tube, so its absorptive abilities are somewhat limited. From the proximal convoluted tubule, whatever's left, so again, we took out all those different things. Whatever's left goes into the descending loop here, the part of the nephron loop that's actually going down. And this part, again, is in the medulla. So up here, we had these upper structures in the cortex, and then down here, these structures are in the medulla of the kidney. And as that water and other non-reabsorbed stuff yet goes down this descending part of the nephron loop, that part of the loop removes water. So as we're going down, the urine is becoming more and more concentrated, and by the time we get to the bottom of the nephron loop, then that urine is probably as concentrated as it can possibly be. So the proximal tube removed a number of things. Descending loop takes only water. As we go up the other side, the ascending loop going back up towards the cortex again, the ascending loop of the nephron actually mostly focuses on reabsorbing salt. So interestingly enough, we have become more and more concentrated as we went from proximal tubule, descending loop of the nephron, very concentrated. As we go back up the ascending loop of the neutron, it becomes less concentrated. It becomes very dilute, actually. And then in the distal convoluted tubule, so we're in our last squiggly part here, we're going to have, again, mostly salts reabsorbed there. And once the urine goes into the collecting duct here, into this tube, it is probably really dilute. And then the collecting duct here actually makes the final decision on how much water to let go in the urine and how concentrated to actually make that urine. So in a way, we went from very concentrated to less concentrated. Now we're going back to more concentrated again. And this seems like we're going back and forth and back and forth, but at each step along the way, we're pulling things back out of the filtrate. I'm not calling it urine yet. We're pulling things out of the filtrate, so the overall volume of filtrate keeps going down. We just adjust the concentration by taking out one thing and then taking out something else further along in the system. So overall, we have probably 80% or so reduction of the total volume of urine or filtrate at this point, which is almost to the point of being urine. So as it goes down that collecting duct, down deep into the medulla of the kidney, we're looking at reabsorption or secretion of salt and water. So if we need to dump water, we can add water to the system. If we need to dump salt, we can add salt to the system. If we need to reabsorb either one of those, we can do that here. So again, the collecting duct really has the final say as to exactly how concentrated that urine is when we release it. And once it runs out of the collecting duct into the kidney, into what's going to be the renal pelvis, and if we go back here, let's take a look at the kidney structure overall. So the renal pelvis would be down here. So we were filtering up here. The collecting ducts were running down the medulla here, and then it dumps into this area. And once it's in this area here, then it's actually considered urine at that point. And it is whatever concentration it is, and it's going to stay that way. So at that point, nothing else happens to it other than just running down the ureter to the bladder to be stored. Now, what we've been looking at is a generic structure of a glomerulus, nephron loops, proximal and distal tubules, collecting duct, all that good stuff. But there are actually two different kinds of nephrons. And what we've talked about so far were considered cortical nephrons. And cortical nephrons have short nephron loops. What we're looking at here looks pretty much the same as we've already seen, but what we find is that the nephron loop on a juxtamedullary nephron, which is what we're looking at here, is much longer. And having a longer nephron loop also means you have a longer collecting duct to go along with it. And this really allows for concentration of urine to an extreme. About 80% of all nephrons in the kidneys are the cortical nephrons. So they can make urine, they can make it reasonably concentrated, but juxtamedullary nephrons, making up the last 20% of the nephrons, are really focused on making a potentially very high concentrate urine. And so that's really their specialty, is really increasing that concentration of urine because they had a lot of nephron loop to remove things and really reduce that total volume of filtrate. And then the long collecting duct along with it takes that very small volume of stuff and can very much concentrate it. So if you're running low on water, probably you're going to find the juxtamedullary nephrons playing a little bit bigger role, as they're still producing urine, but a much more concentrated form. So that's the general idea of urine production, and we really could have spent another couple of hours going through the fine details of things and a lot of technicalities of things, but that's a little bit beyond our pay grade. So hopefully you appreciate now that making urine is a sort of a back and forth, complicated process, but it gets done. And as long as you have adequate water available in the system, then the kidneys will get enough of that waste out. They'll keep that waste flushed out of the system, and things should work pretty well. So hopefully you learned a lot here and it made sense. Thanks for joining us. We'll see you next time.